You are listening audio paper series with Yasir, and today's guest is Petros Zafeiriou from Quorum Institute, Norwich, UK, to discuss his publication, Mining the Watkins Wheat Landrace Collection for Variation in Starch Digestibility Using a New High-Throughput Assay. It is published in Foods Journal. Welcome, Petros.
Hello, Yasir. Thank you for having me here.
Thank you for joining us here. Would you like to introduce us to the background of this study?
Yeah, so starchy foods are the main source of calories for most people, and long-term overconsumption of highly digestible starches can actually impact blood glucose response, which this can lead to different diseases like type 2 diabetes. So at the moment, there are only two ways you can affect starch digestibility. So one is by modifying no genes on wheat that actually they have an effect of changing starch digestibility, or by processing. So both of them are effective, but also they have their drawbacks. And at the end, it all comes down to costs. So for processing, it means basically higher electricity or transportation costs. And for genetic aspects, so if, for example, we know a gene that has an effect on the starch digestibility, it is usually associated with some negative effect on yield. So in our study, we wanted to explore alternative ways. So we focused on exploring more phenotypic variation on starch digestibility by screening natural wheat varieties that have been grown in different parts of the world. And we assume that these actually they will present a wide variation in the starch digestibility.
Wonderful. You explored the alternate way of finding starch digestibility. So how did you do that?
So we chose to screen the core Watkins collection, which actually consists of 118 wheat lines that these capture the variation from more than 800 wheat landraces. Because though most of the in vitro assays can screen up to 6 samples at a time, we had to develop a 96 format, which actually took us more than a year to develop and validate. Also, in our screening, we cooked the wheat samples and we cooled them at room temperature to simulate more realistic approaches to consumption, because we think this is a more holistic approach for finding lines with low starch digestibility.
Lovely. So you developed a method of high throughput screening that can screen 96 samples. What are your findings using this high throughput screening method?
Yes, so there are two main findings that we found in our study. So one is that we validated our high throughput model that can run up to 96 samples with an assay that actually correlates quite well with the glycemic index. And the second main finding was that we discovered that the core Watkins collection has a wide variation in the starch digestibility.
So in this paper, you describe a new method of high throughput screening, and this collection you screened has high variability in glycemic index. What are the applications of this knowledge?
Yeah, so one application could be actually for other research institutes. Now they have the ability to use the high throughput method that we developed, so they can use it as a screening tool for more germplant resources, either on wheat or on different crops. Mm-hmm. And the second application could be so we saw that the Watkins, the core Watkins, we have evidence now they has a wide variation of starch digestibility. So breeding companies can actually take the advantage now to use these resources and develop healthier future wheat varieties.
Thank you very much for sharing very interesting findings with us.